The cell membrane, also called the plasma membrane, acts like a smart security gate around the cell. It is made of a phospholipid bilayer. Think of it as a thin oil layer, hydrophobic fatty tails inside, with water heads outside. Small, nonpolar things can slip through this oily layer easily, but most important biological molecules cannot. 1. Simple passive diffusion, no help needed, no energy used. Some tiny or fat-loving molecules can cross the membrane by themselves just by dissolving in the lipid, oil, part. Examples: oxygen and carbon dioxide gases we breathe. Lipid-soluble things like steroid hormones. They move down their concentration gradient, from high concentration to low concentration, like water flowing downhill. No proteins are needed. It's completely passive. The cell doesn't spend any energy. 2. Facilitated diffusion, help from proteins, still passive, no energy used. Many important molecules are either too big, charged, or polar, water-loving, so they cannot dissolve in the oily middle of the membrane. They need special transport proteins to cross. This is still down the concentration gradient, high to low, so no energy is used. There are two main types of transport proteins for this. Channel proteins like open tunnels or gates. They allow fast movement, especially for small ions, sodium, potassium, chloride, hydronium. Many are selective, only allow certain size and charge ions. They are not always open, some open only when a signaling molecule binds, ligand-gated, like doors with keys, or the electric charge across the membrane changes, voltage-gated, like voltage sensors. Carrier proteins like revolving doors or ferry boats. They bind to a specific molecule, for example, glucose, amino acids, nucleosides. Change shape, and drop it on the other side. Slower than channels. Facilitated diffusion happens down the gradient, just help to cross. Here are some visuals of facilitated diffusion with channels and carriers. Third point. 3 main types of transport proteins. Proteins are grouped into 3 categories based on how many substances they carry and in which direction. Uniporter carries one type of molecule or ion at a time. Usually does facilitated diffusion, down gradient. Example: glucose transporter, GLUT proteins. Symporter, cotransporter, carries 2 different molecules in the same direction. Often does active transport, uses energy indirectly. Antiporter carries 2 different molecules in opposite directions, like a swap. Often does active transport. Here is a clear comparison diagram. 4. Active transport needs energy, goes uphill. Sometimes the cell needs to move molecules against their concentration gradient, low to high, like pumping water uphill. This requires energy from ADP. Primary active transport uses ADP directly. Famous example: sodium-potassium pump, ADPase, an antiporter. It pumps 3 sodium ions out and 2 potassium ions in using ADP. This keeps sodium ions low and potassium ions high inside the cell, very important for nerve signals, etc. Here is the sodium-potassium pump diagram. Secondary active transport uses the gradient created by primary pump, no direct ADP. Example: sodium-glucose symporter, in intestine, kidney. Sodium ions flows down its gradient, into cell, and pulls glucose against its gradient, into cell 2. Here is the glucose-sodium symporter diagram. So, that's all for today. I hope you understood it. We will meet again in the next video. And please subscribe to my channel and also press the bell icon.